Welcome to PHU classes. Today we will learn what is motor neurons and muscle spindles.
In order to do the forward bend position during your yoga class, your nervous system has to do a number of things. First, an upper motor neuron from your brain, specifically your cerebral cortex, has to send a signal down to a lower motor neuron that's in the anterior horn of the spinal cord. This lower motor neuron is also called an alpha motor neuron, and it relays an action potential through an axon that goes to muscles in your legs, which enable you to extend them. Now, when you stretch or flex your muscles, proprioceptors that detect the position and movement of the muscles initiate reflexes that prevent you from damaging the muscles from overstretching or overcontracting. These proprioceptors are scattered throughout your skeletal muscles and operate on a subconscious level so you never even notice them. Now, a muscle is made of a bundle of muscle fibers with extrafusal muscle fibers on the outside and intrafusal muscle fibers on the inside. Extrafusal muscle fibers provide most of the force during a muscle contraction and are innervated by lower motor neurons, which are also called alpha motor neurons. Extrafusal muscle fibers attach to bones with tendons, which are a specific type of connective tissue. These tendons have proprioceptors called Golgi tendon organs, which lie at the ends of these extrafusal fibers. Now, if we pull apart the extrafusal fibers, there's another proprioceptor called the muscle spindle that lies within the extrafusal fibers. Each muscle spindle contains multiple intrafusal muscle fibers. Just like extrafusal muscle fibers, intrafusal muscle fibers have contractile proteins like actin and myosin. However, these contractile proteins don't extend through the entire length of intrafusal muscle. Instead, they're only present at each end of an intrafusal muscle fiber. Therefore, the central region of an intrafusal muscle doesn't contract, even though the ends do. The central portion of the intrafusal muscle fiber contains the muscle fiber's nuclei, and the arrangement of the nuclei determines whether the intrafusal muscle fibers are considered nuclear bag fibers or nuclear chain fibers. Nuclear bag fibers are ones that have their nuclei concentrated in a wide, central portion of the fiber, like a sack full of oranges. On the other hand, nuclear chain fibers are half as long as nuclear bag fibers, and have their nuclei arranged in series like peas in a pod through the central region. Coiled around the central region are two kinds of sensory neurons that depolarize when the intrafusal fiber is stretched. Type 1A neuron fibers coil around the central region of both the nuclear bag and the nuclear chain fibers, and relay information about how far and how fast the muscle is being stretched. Type 2 neuron fibers branch out and attach to the ends of the central region of the nuclear chain fibers, and relay information about how far the muscle is being stretched. In addition, gamma motor neurons innervate both ends of the intrafusal muscle, which contain actin and myosin. And when stimulated, the gamma motor neurons cause these end regions to contract, which shortens the intrafusal muscle and keeps it taut. First, let's look at the stretch reflex, which prevents overstretching of the muscles. So let's say you're sitting on a high stool with your legs comfortably dangling. In that position, the extensor muscles in your legs, like the quadriceps femoris, are at their resting length, and the type 1A and type 2 neuron fibers are firing off action potentials at a normal rate. These action potentials travel through the peripheral nerves to enter the dorsal ganglia, where the cell bodies of both types of fibers are located. Each cell body also gives off another axon that continues on into the spinal cord. Type 1A fibers go to the anterior horn and directly synapse with alpha motor neurons of the muscles being stretched. Type 2 fibers also go to the anterior horn to synapse with inhibitory interneurons, which synapse with alpha motor neurons of the antagonist muscles, or muscles that serve the opposite function. In this case, they're leg flexors like the hamstrings. Now, if you tap the area below your patella, where the quadriceps femoris muscle attaches, it stretches the muscle tendon a bit. This also stretches the extrafusal muscle fibers and the muscle spindles. Normally, they only get stretched when you're flexing your leg, so it's like sending your muscle spindle a bit of false information. So what happens is that the stretched muscle tendon causes an increase in the rate of the action potentials being fired by the muscle spindle. That signal travels through type 1A and type 2 sensory fibers, which relay the message to cells in the spinal cord. Type 1A fibers synapse with alpha motor neurons of the leg extensors and cause these muscles to contract, which prevents damage from overstretching. At the same time, the type 2 fibers trigger the firing of the interneurons, which inhibits the alpha motor neurons of the leg flexor muscles, which cause them to relax. As a result, the leg kicks out and the extensor muscles are shortened. This whole process is called the patellar reflex, or knee-jerk reflex, and it's involuntary since the afferent and efferent signals never need to go up to the brain. So when the muscle spindles are stretched and get longer, it triggers a reflex. Now, when the muscles are contracted, the muscle spindles are shorter, and this is where the gamma motor neurons play a role. The brain sends a motor signal down an upper motor neuron to stimulate the lower or alpha motor neuron in the anterior horn. The alpha motor neuron travels to the extrafusal muscle fibers, causing them to contract and shorten the muscle, and the muscle spindle becomes slack. As a result, the muscle spindle slows down its rate of firing action potentials. But the key is that while all this is happening, the upper motor neurons also send a signal to the adjacent gamma motor neurons in the anterior horn. Gamma motor neurons cause the contraction of the intrafusal muscle fibers in the muscle spindle, making them shorten as the muscle shortens. This is called alpha-gamma coactivation, and maintains the tension of the muscle spindles and keeps them sensitive to stretch even when your muscles are contracted. So the stretch reflex prevents damage from overstretching. Next, let's look at the Golgi tendon reflex, which prevents damage from overcontraction of the muscles. The Golgi tendon organs are proprioceptors like the muscle spindles, and they're found in the tendons. Now, let's say you go to a plant nursery and pick up a large potted plant. When you contract the biceps brachii muscle, the muscle shortens as tension is generated. The Golgi tendon organ in the biceps tendon gets compressed as the tendon shortens, and that makes it fire off more action potentials. These action potentials travel through a type 1b afferent fiber through the dorsal root ganglion, which contains their cell bodies. Once they enter the spinal cord, they synapse with inhibitory interneurons, which in turn synapse with alpha motor neurons of the contracting muscle, in this case the biceps brachii. At the same time, 1b afferent neurons also synapse with stimulatory interneurons that synapse with alpha motor neurons that innervate the antagonist muscles, in this case the triceps brachii. So we get relaxation of the biceps and contraction of the triceps, which limits the tension generated in the biceps muscles and prevents them from being damaged. Alright, as a quick recap. The muscle spindles and Golgi tendon organs are proprioceptors that detect the change in muscle length. The muscle spindles trigger the stretch reflex, where an overstretched muscle spindle sends afferent signals through type 1A and type 2 sensory neurons to the spinal cord. 1A sensory neurons cause the contraction of the muscle, and type 2 causes the relaxation of the antagonist muscles. The Golgi tendon organs trigger the Golgi tendon reflex when a muscle is being overcontracted. It sends afferent signals through type 1b afferent fibers to the spinal cord, which triggers the inhibition of the contracting muscle and the contraction of the antagonist muscles.
Thank you.